All right, so it looks like the number is starting to stabilize. Then, oh, someone raised their hand. Hi, Paula. Then I guess we can get started. Hey, thanks everyone for being here. We're very excited to see you. So first I'm gonna hand it over to Erica to do the land acknowledgement today, and then we'll get started.
Thanks, Kate. Hi, everyone. My name is Erica Bush, and I'm a second-year graduate student at Yale University. And thank you all for joining us this afternoon for the next talk in the Innovators in Cognitive Neuroscience speaker series. And before beginning, we want to acknowledge the Indigenous lands from which we're joining today's talk. So I'm speaking to you from New Haven, Connecticut, which was land taken from 3 Indigenous peoples, the Quinnipiac, Paugussett, and Wappinger peoples. And please use the link that I'm about to send to the chat to research and reflect on the Indigenous lands from which you're joining us. And through the acknowledgement of past harms, we step towards a more equitable and humane future. So on behalf of all the students and postdocs on the organizing committee, I'm excited to welcome you all to today's Innovators in Cognitive Neuroscience seminar. The organizing committee together represents a consortium of colleges and universities, including Dartmouth, UPenn, Yale, MIT, Princeton, Harvard, Columbia, and Gallaudet, that are committed to highlighting advances in the cognitive neuroscience research. We acknowledge the privilege of these institutions, and we wish to make the science discussed here available and accessible far beyond our ivory tower of academia. And so with the consent of our speakers, we plan to make these talks publicly available on our website, and our seminars open to anybody who are interested. And a quick note to those of you who are new to this series, after the talk is over, we'll move over to a new Zoom room for the Q&A, which I'll post in the chat in one moment. And this will be a space for anyone to ask additional questions that weren't answered during the talk, and or any questions about Dr. Uddin's career as a scientist. And we'll post this link once more after the presentation has concluded. And so next, I'm going to hand it over to Chi, who will introduce today's guest.
Thank you, Erica. Hi, everyone. My name is Chi Lin, and I actually recently got my PhD from Yale. Today, we're very fortunate to have with us Dr. Lucina Uddin. Dr. Uddin received both her undergrad degree and her PhD at UCLA. She then went on to complete her postdoc training at NYU and then at Stanford. In 2014, she joined the University of Miami as a faculty member. At University of Miami, she was the founding director of both the Cognitive and Behavioral Neuroscience Graduate Program and the Cognitive and Neuroscience Division there. In 2021, she returned to her alma mater, UCLA, as a professor and is currently directing the Brain Connectivity and Cognition Lab there. Her lab investigates the relationship between connectivity in large-scale brain networks and flexible behavior in both typical and atypical development. In particular, she has pioneered the study of the role of insula and the salience network in higher-level cognitive processes and how these systems may differ in neurodevelopmental disorders. I could go through her CV and list all of her amazing accomplishments, but then you probably won't get to hear from Dr. Uddin herself in the next hour. So I'll save you all from that. I'll just highlight a few of the honors that she's gotten, including the including the inaugural Diversity Inclusivity Champion Award from OHBM, the CFAR Azrieli Global Scholar Award, the US URN Prize, the OHBM Young Investigator Award, and many others. Before I hand it over to Dr. Uddin, I also just wanted to quote a colleague of mine that has worked with her before when speaking about her experience when working with Dr. Uddin, which truly shows beyond being a rigorous and world-renowned scientist. Dr. Uddin is also an amazing mentor and leader of the field. So here are my colleagues' words. Lucina is the most wonderful mentor ever, exclamation mark. She's such a badass, never afraid to speak her mind and disrupt the system. She's super committed to diversity and inclusivity efforts, 2 exclamation marks. She emails back at lightning speed, hosts the most epic lab happy hours, often with sushi boats, and forms amazing collaborations all around the world. End of quote. So with that, please join me in welcoming Dr. Lucina Uddin.
Wow, that is a very hard intro to follow. Thank you so much for this really warm welcome. Thank you to the anonymous person who I think I can guess who that was, who the quote was from. Thank you. It's really a pleasure to be here today, and I really enjoy the idea behind this Innovators in Neuro Forum, and I appreciate all the work that you're doing to put together this initiative. I'm really happy to share with you some of our work over the last few years on brain dynamics and flexible behaviors. And I'm really thrilled that I'll get a chance to chat with some of you after this as well. So our lab is really interested in studying the brain basis of flexible behaviors for lots of reasons, as we've all learned how important flexibility is now during the time of the pandemic, where we've switched from everything we've done to going to Zoom for our talks and our lectures and most of our socializing for 2 years. So we know what being flexible is. But what we're in our lab, we're often interested in this condition, autism spectrum disorder, and children who are diagnosed with autism often have difficulties in everyday flexibility. They might want to wear the same pair of socks every day or eat a particular type of food. And this insistence on sameness is one of the characteristics that is really challenging for caregivers and educators and people who are working with individuals with autism. So it's something that we've been interested in for quite some time. But very broadly, our lab is studying how brain networks develop across the lifespan, from childhood to adolescence and adulthood, and what are the consequences of atypical development across and within large-scale brain networks. So, and we're of course interested in how these network developmental processes relate to cognitive development. Oops, I can't see my top of my slide now, but okay, we'll do without it. So what we do is we study groups of typical adults, typically developing children, and clinical populations in parallel using multimodal neuroimaging approaches, functional MRI, diffusion, and structural MRI, as well as causal connectivity modeling. And we try to kind of do these studies in parallel so that they can all inform each other. And in our the back of our minds, we're always thinking of these foundational principles of network neuroscience. The idea that cognition is subserved by interconnected networks and behavior is mapped at the level of multifocal neural systems rather than specific anatomical sites. And this is a nice quote from a paper by Marcel Messlin back in 1990, really before the advent of modern neuroimaging techniques. We still had this idea that you can't sort of easily localize function from one brain region to one function, but usually it's multiple regions and working in a networked fashion that underlies anything that we do that's complex. And I think there's also some really nice ideas from the computational neuroscience literature. I'm just pulling out one idea from Randy McIntosh, where he talks about the idea of neural context. And here the quote is that the functional relevance of a given area depends on the status of other brain areas. So you can imagine that a network colored in gray here might behave differently in the context that you see in A versus the context in B, where there's different co-activated nodes. So I think this is also a nice sort of thing to remember in all of our approaches to cognitive neuroscience. And lately, we've also been very interested in this idea of dynamics and understanding changes over the course of seconds and minutes in the brain, and how for us, really, this is a nice quote from Pessoa about how a network needs to be understood in terms of interactions between multiple brain regions as they unfold temporally. So we've taken this idea of dynamics very seriously. And one discovery that we're all now familiar with back in 1995 by Bharat Biswal is that even in the resting state, our brain is still cycling through these different patterns of activity in a spontaneous way that is not random, but actually coherent within functional systems. So if you stare at a resting brain long enough, you'll see that it tends to cycle between networks that are important for language, memory, attention, all of the processes that we do. It turns out our brain cycles through these systems at a very low frequency, even when we're not engaging them at all. So of course, we and others have exploited this phenomenon to understand the integrity of large-scale brain networks and how they change across the lifespan and in clinical populations. So over the course of the pandemic, because of all the time we've had away from the lab, I had a chance to review some of the literature on flexible behaviors and cognitive neuroscience, and gave me the opportunity to really think, across different paradigms and fields, how we think about flexibility. In humans, we often use tests like the Wisconsin card sort test to index flexible behavior. In animals, there's a reversal learning tasks that require rats to switch between different stimulus outcome mappings, and that's one way of assessing flexibility. And then, of course, when we go over to developmental psychology, we might make some cuter stimuli in order to, ask questions like, press one button when the fish is facing one direction, or press one button when the fish is this color, and get people to switch between different tasks. And that's another sort of way we've indexed flexibility. Let's see if my slides will go forward. We've figured from all of these studies that there's frontoparietal and frontostriatal and cingulo-insular brain regions that tend to be really involved in all aspects of flexible behaviors. And I think it's interesting to study flexibility trends diagnostically, because there's many disorders across the lifespan that involve deficits in flexibility, starting from autism and ADHD in early life, and going through things like depression and anxiety, which involve repetitive negative thinking or rumination or getting stuck on certain thoughts, as well as OCD, of course, obsessive compulsive disorder. Schizophrenia is involved with the excessive cognitive rigidity, and late life dementias, including Alzheimer's and Parkinson's, are also characterized by rigid thoughts and behaviors. So I think there's some reasons that we could study flexibility in all these different conditions, and we could learn something really fundamental about how the brain works. What we've done in the lab is take a study or a task that was developed for children in the developmental psychology literature called the flexible item selection task, and adapt it for use in the MRI environment. So here's a figure showing what a child or a participant would see. And you would ask them, pick two things that go together. And they might say, well, the second and the fourth are both blue. And then you'd say, okay, well, pick two other things that go together in a different way. And they might say, well, the third and fourth are both rabbits. And you could go on and ask for different kinds of groupings. And of course, this would require flexibility to switch between different stimulus dimensions. So putting this in the scanner, we came up with a control condition where participants are just asked to press the buttons corresponding to the boxes that are outlined. So they would, press these boxes in this order. And this is a task that Dina Dajani, a former graduate student in the lab, really took a long time to develop, and did a lot of work creating the stimuli, putting it in the scanner, testing this on adults first to see if we got expected patterns of brain activation in the flexibility minus control trials. And she spent a lot of time coming up with accuracy and reaction time indices, checking that the task is, the behavior is similar between the scanner and in the scanner and outside of the scanner. So a lot of work went into just validating this task for several years. But we found what you might expect, really, really strong, robust activation in frontoparietal brain regions, cerebellum and basal ganglia, and anterior cingulate and anterior insular cortices. And this was really basically strong activation patterns when people are engaged in this type of flexible item selection. And the reason we spent all this time was, of course, we wanted to bring something into the scanner that children could do and that children with autism spectrum disorder could also do to let us tease apart the brain mechanisms underlying inflexibility in those conditions. So in another line of work that I won't talk about in the lab, we spend a lot of time looking at lifespan trajectories of different brain measures like signal variability, how that changes between the ages of, for example, 6 to 85. We spend some time also looking at, looks like it flipped ahead. Spend some time looking at structural connections as they change between childhood and adulthood, effective connections or causal connectivity between children and adults, differences in network connections across the whole brain, how some become more connected and some become less connected, and the spatial topography of networks across the lifespan. So this is sort of a backdrop of a lot of the work we do in clinical populations, just understanding typical developmental trajectories. But as one might expect, children are able to do this task, and they're able to engage frontoparietal and singular-insular and some of the same systems that we see adults use when they do this task. And Lauren Kupis is the one in our lab working on this registered report right now. But the bad news is that as we were conducting this study, we came upon March 2020, and that was when all scanning ceased to exist. So we were not able to complete all the data collection that we had planned. And so we have a smaller group of children with autism that Lauren is currently analyzing and hoping to find some interesting signatures in that group. But this is a study that's basically not at its conclusion, and perhaps never will be, because a lot of things have changed since March 2020. But in any case, what I wanted to summarize is that cognitive flexibility, we think, involves coordination among brain regions supporting executive functions broadly. And groups of adults and children seem to be engaging lateral frontoparietal and mid-singular insular networks during flexible item selection. And I think I lost some slides in there. Well, anyway, so I had a slide about task-modulated connectivity of the inferior frontal junction that was important for flexible behaviors. And so perhaps we'll be able to show you some shortened or abbreviated fMRI data collected from children with autism in the future. So I'll start or go back to a little bit some of our work in typical adults, looking at the brain basis of flexible behaviors. And we've been able to use some of these new approaches for looking at dynamic functional connectivity. So instead of saying, okay, across 10 minutes on average, how are different brain regions talking to each other, we can actually break the time series down into 40 or 60-second windows and say, okay, moment to moment, how are these whole brain connections changing? And you can use approaches like k-means clustering to look at different occurrences of brain states, and you can calculate a lot of metrics, like, for example, how frequently does one particular connectivity profile occur, or how long, or what's the dwell time once you get into one functional connectivity state, how long does that persist? And you can also measure transitions between brain states, so how many times do the switches between brain states occur? And so Jason Nomi in the lab has spent a lot of time working on the Human Connectome Project data, and you can find regions of interest that the brain's already parcellated. This is a study from a few years ago where you just used a couple hundred subjects. But the nice thing is that there's 1 hour of resting state fMRI data from these subjects. So you really have the large, long data set that you need in order to do these dynamic analyses. And of course, these participants have also done a whole lot of neuropsych testing and behavioral testing outside of the scanner. So we have a lot of information about their individual differences in performance on things like the Wisconsin card sort task and other tasks. And what we find overall is that the brain seems to toggle between different states, 5 here that I've shown ordered from most frequently occurring on the left to least frequently occurring on the right. And what's most interesting really is that those individuals who seem to spend or show more frequent occurrences of state 1, which is characterized by loose connectivity between brain regions, and less of state 5, which is a highly interconnected state, those are the ones who seem to do better on working memory. And similarly, the individuals who show more occurrences of state 2 and less of 3, again, the loosely connected states, those are the ones that are associated with better performance on the Wisconsin card sort or cognitive flexibility outside of the scanner. So this suggests that greater cognitive flexibility in adults is associated with the propensity to occupy more frequently occurring configurations that are characterized by attenuated correlations across the brain and greater variability of those correlations. And this is just one example of how a dynamic functional connectivity approach can help us to understand relationships between whole brain dynamics and flexible cognition. So, of course, what I've shown you up until this point has all been whole brain, not specific to any systems, but other work in our lab has really focused a lot on 3 networks that show up ubiquitously in cognitive neuroscience across tasks, across different studies. They go by different names. I'm trying to standardize some of these network names, but the idea is that you see oftentimes regions that are described as the default mode network or medial frontoparietal network with key nodes in ventromedial, prefrontal, and posterior cingulate cortex involved often in internally directed processes. And on the other hand, the lateral frontoparietal systems, sometimes executive control networks, involved in more externally directed goal-oriented actions. And then the mid-cingulate insular or salience network, which is here in blue and involving the anterior cingulate and anterior insular cortex. And there's a lot of functions that I think are really important that this network serves. But one thing we've noticed over the years is that if you do any kind of causal modeling, you can use the signals in the insula to predict what will happen at later time points in the default and the central executive, which is why we think it plays a role in switching between activity in those 2 other networks. And so using this kind of network framework, we've looked at development of cognitive flexibility across the lifespan and how it relates to dynamics among these 3 networks. And so this is, again, work that Lauren Kupis published last year. And she was able to take advantage of a large data set collected by the Nathan Klein Institute. Here there's over 600 subjects in this analysis, resting state fMRI subjects between the ages of 6 and 85. And these participants have also completed cognitive flexibility tasks outside of the scanner, like this trail making task, which I'm told is very difficult and is a nice challenge. And what she did actually is use a different dynamic connectivity approach known as coactivation pattern analysis, which looks time frame by time frame and groups and finds areas of activation surpassing a certain threshold and groups those together. But the point is here that we don't use sliding windows, but we can still calculate things like transitions between brain states and frequency of occurrence of brain state and dwell time of certain brain states. And so what Lauren was particularly interested in is looking at key nodes of the salience, the default, and executive network, and looking at the dynamics of those 3 systems across the lifespan, first just to characterize how they change. And what's interesting is that a pattern of coactivation involving default and executive, so that's when those 2 networks are both active at the same time, that is a coactivation pattern that shows a highest frequency of occurrence in mid-age, and then lower in early life and lower again in late life. So it shows this nice inverted U-shaped trajectory, which is consistent with some models of aging, like the default executive coupling hypothesis of aging by Nathan Spring and colleagues. And also, interestingly, the number of transitions between brain states changes across the lifespan in ways that differentially relate to cognitive flexibility. So this is a complex interaction here, but basically the individuals in sort of midlife, the number of switches between brain states does not really relate to their performance on the trail making task. But for younger and older individuals, so early life and then late at the other end of life, if you have more switches between brain states among these 3 networks, you do better on the switching task outside of the scanner. So there's something dependent about your developmental stage that interacts with the, how much you need brain state transitions in order to accomplish a particular task here. So here, this is just to summarize the frequency of coactivation patterns involving lateral frontoparietal and medial frontoparietal networks changes across the lifespan. Oops. And the brain state transitions support cognitive flexibility in different ways at different stages of the lifespan. So since I spent so much time focusing on the insular cortex here and what I think it's doing, I think I'll just spend a minute or two just introducing some of its functional anatomy. We know it's involved in studies of emotion, empathy, and pain, but also very much implicated in subjective awareness interoception and somatosensory processes. And at the same time, high-level cognitive processes like conflict, switching, attention, and inhibition seem to all engage the insular cortex. So it's kind of become my favorite brain region because it seems to be able to do everything. But if you look closely and look at parcellation studies that try to subdivide the insula into subdivisions, basically, with different functional connectivity profiles, there's at least a few different studies that have done this. And here I'm showing a few from Ben Dean and Luke Chang, where they've used resting state fMRI to subdivide the insula by basically clustering by patterns of whole brain functional connectivity. So they have found evidence for at least 3 subdivisions, and I'm sure there's many more depending on which study you look at, but there's at least a dorsal anterior insula, a ventral anterior, and a posterior insula based on some of these parcellation studies. And if you look at meta-analyses and coactivation kinds of analyses, you can find here some differences in these subdivisions in terms of which tasks really engage them. So on the left, you can see this is a figure from Luke Chang's paper. When the ventral anterior insula is active, it's usually tasks that use the terms there in red, like emotion, anxiety, kind of affective types of processing. And on the right, I'm showing a coactivation meta-analysis that I conducted some years ago in collaboration with Luis Pessoa and Michael Anderson, where we showed that across tasks, when the ventral anterior insula is active, the other areas of the brain that tend to be coactive are limbic regions. And the dorsal anterior insula seems to be showing up when there's studies of inhibition, switching, error processing. And on the right, you'll see that the frontal parietal cortices tend to be coactive whenever the dorsal anterior insula is active. And the posterior insula seems to be showing up more in studies of pain and somatosensory and stimulation. And when you see the posterior insula active, the coactivity tends to be in those temporal and sensory motor types of regions. So, but at the same time, all of these subdivisions do seem to be quite active and converge across all different task domains. So it is sort of a does everything kind of brain region in some ways, even though sometimes we see these subdivisions acting in specific areas, specific domains. So one thing we wanted to do is just see if this had this functional complexity of the region could somehow be explained by dynamics of how the areas interact with the rest of the brain. So this was when Jason Nomi came in and did a sliding window analysis using different subdivisions of the insula. And it's really probably impossible to read, but again, we found 5 different connectivity profiles where in some of those states, the different subdivisions of the insula had similar functional connectivity patterns to the rest of the brain, like for example in state 3. Whereas in state 2, which occurs very infrequently, you see that the insular subdivisions diverge in their whole brain functional connectivity patterns. And I actually think the most interesting finding from this study, which will hopefully pop up in a second, is that if you look at the dorsal anterior insula, that's the one that shows the most flexibility itself in terms of that's the brain region that is the most different between its different connectivity states. So it sort of interacts more flexibly with different brain regions compared to the other subdivisions of the insula, which show pretty similar patterns of functional connectivity with the rest of the brain, regardless of which connectivity state they're in. So we had a great undergraduate student, Will Snyder, in the lab, who's now working at Cambridge, and basically was able to look at these dynamics across the lifespan. And this was a nice paper that he just published last year, looking at the different connectivity states and how they change in their frequency of occurrence from, between the ages of 6 and 85. And he found just one finding I'll highlight here, that there was a state in which the salience network, including the insula and ACC, was kind of isolated from the rest of the brain, and that it wasn't as talking or as connected with other brain regions. And he was able to use that same data set I described before, 600 subjects, resting state fMRI data, just to show that there's, again, some lifespan trajectories where that salience network isolation, I guess, if you want to call it, it shows up or persists longer in early life and in late life, but it doesn't dwell as long, or we don't see that state as often in midlife. So there's a lot of kind of things I'm glossing over here in the interest of time, but I'm happy to chat about later on. But we've also looked at structural connections of the different subdivisions of the insula and found there's some unique ways in which the dorsal anterior subdivision talks to the frontal and thalamic and subcortical regions that we think might also be involved in its sort of ability to do so many things. So basically, we think the insular cortex in adults can be parcellated into at least a dorsal, a ventral, and posterior subdivision, with the dorsal more involved in higher level cognitive control, ventral more involved in emotion and affect, and the posterior more involved in sensation. And we think that the singular insular or salience network dynamics show some interesting and complex maturational changes across the lifespan. And the dorsal anterior insula seems to be the one subdivision that exhibits the most variable functional connections and also appears to have unique structural connections that we think might enable its functional flexibility. And just incidentally, that was also the subdivision that we found was the one that causally influences the default and executive networks. Although I wasn't looking at subdivisions at the time, it turns out that the other subdivisions don't show that same kind of causal effect. So there's something really specific about the dorsal subdivision. Now, of course, we're using fMRI where you can't, you have a sluggish response and we can't get millisecond resolution. But a lot of groups have now started using intracranial recording from subjects who are being monitored for epilepsy in the neurosurgical wards. And there's been a lot of nice recent work by other groups who have tried to understand network dynamics at the millisecond scale using a lot of these surface electrodes. I have an ongoing collaboration with colleagues at the University of Western Ontario, and their neurosurgical unit is one of the few that plants depth electrodes in the insular cortex. So this affords the opportunity to look at the different subdivisions I mentioned, the anterior, ventral, and the posterior, in a way that hasn't been done before. So this is another area where we're actively looking to collect data. And although we started this project before the pandemic, it was delayed like everything else. So I do hope that we'll be able to get some data from these really unique participants and be able to learn a lot more about the timing and the interactions between these regions and the functional specificity of the different insular subregions as we get further and further into this project. So in the remaining minutes, I will be able to touch a little bit on how all of this work that we do in typical adults and in typical development and in lifespan trajectories helps inform our studies of clinical populations, where we focus a lot on autism spectrum disorder, but there's some other areas as well, but I'll just share some of the autism work today. So in autism, there's the social communication deficits, I think, grab the most attention, and there's a lot of interventions that are tailored for bolstering those issues. But one of the things that's less well studied is flexibility, or sort of the difficulties in flexible behavior in autism. And I think they might be related to these grim statistics that we often see, which is that individuals with autism often don't live independently. And this is a surprisingly low number compared with other developmental conditions that are also occurring in early life. And similarly, there's low rates of employment or high unemployment in young adults with autism compared to some of these other disabilities. And I think that the flexibility deficits have a lot to do with this. Of course, social communication is a main issue as well. But if you're trying to take the bus to get to work and the bus never shows up, then you have to flexibly come up with a new way of getting to work. And those are just one mundane example of flexibility in daily life. But that's kind of what happens when you're living independently and working on your own, is you're constantly making these judgments that require some degree of flexibility in order to to perform your job. So I think this can be a challenge for those with autism. And there are some treatments that really specifically focus on things like enhancing flexible behaviors. One of them here is called Unstuck and On Target, that targets flexibility specifically. And what's really not known yet is which children benefit the most from these kinds of treatments. And part of that is because of the heterogeneity in autism. So we do see that there is across the board heterogeneity across symptoms and across cognitive profiles. So, one of our students, again, this was Dina's work, did a study some years ago looking at executive function profiles in a mixed group of children. These were, some children had autism, some had ADHD, some had autism and ADHD, and some were typically developing. And this was a collaboration with Stuart Mostovsky's group at Kennedy Krieger. And she found that using latent profile analysis and behavioral indicators of executive function that there was evidence for at least 3 classes of impaired, above average, and below average, average, impaired, and above average executive function classes, and that the kids with disorders didn't necessarily fall into the impaired executive function class. In fact, if you look at this pie that shows kids that had average executive function, so no real deficits, some of those kids were those with autism, some were the ones with ADHD. So it's not as if just having the diagnosis automatically means you'll have a problem with executive function. And one of our undergraduates in the lab, Adriana Baez, did a follow-up study and published this as well as a replication to show, again, that basically you can have different profiles of executive function across neurodevelopmental conditions, but it's not necessarily the case that having autism will put you in the impaired group. So this heterogeneity is something that, of course, we all are trying to deal with. One of the things that we thought might relate to this individual heterogeneity is the underlying brain dynamics that contribute to flexible behaviors. So by now you might imagine that we'd have some hypotheses. For example, perhaps the kids with autism are just showing fewer switches between brain states, and perhaps that's related to their difficulties with flexible behaviors. So this was something that Jason Nomi in the lab again went back and looked at using some publicly available data from the Autism Brain Imaging Data Exchange. And so he was able to pull some data sets together to look at brain dynamics across the whole brain again here, and did find some initial evidence for a reduced number of transitions between brain states in the individuals with autism compared with typically developing individuals. And again, we wanted to go from the whole brain to individual networks to look at if we could get at more specific areas. And so Emily Marshall was another undergraduate honors thesis student in the lab who published this study. And it's amazing what undergrads can do when you give them data and let them work, basically. And so we've had a lot of undergrads in the lab who've been able to do great things. So this project, where she took basically regions of the salience or mid-singular insular network and used the co-activation pattern analysis method that I mentioned before to look at whether or not children with autism showed different patterns of co-activation involving the system. So she did, in fact, find one pattern. This was a co-activation pattern where the network on the top left was co-active with the regions you see on the bottom. And this particular brain state was showing up less frequently in a large sample of children with autism. So that's sort of some evidence that there's differentially affected salience network or mid-singular insular network dynamics in children with ASD. And here we have a task that we actually took from BJ Casey, and it was developed many years ago as a flexibility task or a set shifting task to be used in the scanner with children. And it turns out here, we just ask a simple question for the child to point out the object that's different. And in some cases, the object will be different because it's a different shape, and sometimes it's different because it's a different color. And it turns out children with autism, and typically developing children in the age range we were looking at, 7 to 12, they're able to do this test pretty well in the scanner. And so what Bryce Dirks in the lab did was he took the data from this task that we had children with autism and typically developing children perform. They were behaviorally pretty much the same. Both groups were able to do the task. So instead of there weren't too many differences in brain activation between the groups. But what we did find is that if you look at different co-activation patterns involving the three networks that I like to focus on, the cingulate-insular or salience, the central executive or the lateral frontoparietal, and the default mode or medial frontoparietal. So if you take nodes of those networks and do co-activation pattern analysis across them, you can identify different time points at which these different networks work together. And in the kids with autism, excuse me, there's one co-activation pattern that involves basically the central executive network that seems like it's engaged more in kids with autism, especially towards the final run of the task. So as they're getting towards the end of the task, it seems that kids with autism need to engage more of these executive networks in order to maintain the same level of behavioral performance as the typically developing kids. So this is all just a tour of how dynamic functional connectivity approaches can help reveal atypical patterns of dynamics that are prevalent in neurodevelopmental disorders characterized by cognitive inflexibility. And of course, the extent to which these dynamics underlie individual differences in flexible behaviors is something that we're currently investigating, and we're hoping we'll have more data on shortly. So all of these studies are sort of looking at brain mechanisms underlying flexibility because we think that this will help us understand more about autism and how we can help kids sort of move away from insistence on certain inflexible behaviors and focus more on their interactions with the world. And this is, of course, work that's not done alone. There's so many individuals who contributed data and code and financial resources and just wonderful collaborations with folks all over the world. So this is kind of work that certainly just cannot be done in isolation. And I wanted to draw everyone's attention to, so we have a research topic in Frontiers that Stephanie Bodison and some other colleagues are spearheading. But the idea is we wanted to launch a research topic focusing on justice, equity, diversity, and inclusion in the neurosciences. And we're open to all kinds of submissions, review articles or empirical papers on any topic that's relevant to JEDI issues in neuroscience. So please do consider submitting something. The deadline is set for July, so there's plenty of time. And of course, extensions are always possible. So please feel free to ask any questions about that. And I'm currently living in Santa Monica and working at UCLA and couldn't be happier. So I am excited to take any questions at this point, and thank you so much for your time. Oh, wait, one more thing. So I wanted to also just mention some of the work that I was doing related to mentoring and diversity with the Organization for Human Brain Mapping and with the Flux Society in terms of mentoring programs and just thinking more generally about how we can revise evaluation metrics, both at the graduate admissions level, removing GRE requirements, and thinking about holistic reviews and things like that for faculty advancement to dismantle privilege. And one of the things I'm really excited about now is just promoting this idea of casual mentorship. So basically, we do rely a lot on formal mentoring networks, but we also often just run into people in the hall and ask a question that's easily answered. But not everyone has the same networks in order to benefit from that kind of thing. And I think in particular, minoritized groups might not necessarily have the same networking resources. So I'm encouraging senior scientists to indicate their availability. Essentially, if you're willing to talk for half an hour with a student, just put it on your Twitter account. I'm available for mentoring, and it's amazing what what just half an hour of chatting at certain career stages can do to help move people along when they don't necessarily have the networks in place in their current institution. So I think that's basically all that I wanted to cover. So I will now I'll be happy to stop and take any questions. So that's funny. This slide didn't show up. I think my slides were skipping, but this is the lab and my collaboration network. And now I'm happy to take questions. Okay.
Great, thank you so much for such a wonderful talk. So for anyone who's new to this talk series, the way we handle Q&A here is that you can post your questions to the Q&A tab, and then Erica and I will take turns to read your questions. Okay, so actually there's already a question there from David DiStefano. Sorry if I mispronounced your name. So I think this question is about probably the intracranial work that you talked about. So his question is, have you discovered much regarding the brainwave frequency bands intrinsic to supporting cognitive flexibility between the three primary networks or microstates? I'm currently researching this in the EEG domain, where it appears to be the upper alpha band that's most pertinent.
Yeah, that's a great question. Thanks for asking. Unfortunately, I haven't worked in the EEG domain, so I haven't been able to test this this question at this point. And we're still waiting to collect some of the intracranial data. So that's been on hold. But yeah, I think it's wonderful to be able to integrate across modalities to get at this question. But of course, the folks who are doing simultaneous EEG fMRI will tell you how difficult that is technically, and also very difficult to do with children and with developmental and clinical populations. And so, yeah, the short answer is I don't know at this point.
Perfect. Thank you. I personally have a question about, because at the beginning when you introduced like the importance of cognitive flexibility, you mentioned like rigid thoughts. So I take it to be more a question about like the difference between being flexible about our internal thoughts, being stuck in like internal brain states, versus like we can also be inflexible in terms of how we react to the environment. So have you like thought about, is there any difference in terms of the networks that's involved in, involving like switch within our own thoughts versus like switch in response to something that's more external?
Yeah, that's a great question, and I think there's definitely a degree of overlap, even though we study them in isolation. We study rumination in depression, and we study worry and anxiety as inflexible thought patterns, and then we study behavior and task-related flexibility in other areas. But I think they're probably all relying on a similar mechanism, because we see a lot of the same brain systems involved in these different types of flexibility, even though they seem different. Like they seem like it's an internal thought process or an external behavioral thing. But I mean, I think you have to have some flexibility in your thoughts in order to behave flexibly. So I think it's all interrelated. And sometimes I think it's just that we use different terminologies in these different fields. Like in the clinical sciences, we talk about things a certain way. In the behavioral neurosciences, with animal work, we talk about things in a certain way. In cognitive neuroscience, with fMRI, we talk about things in a different way, even though I think there's reason to believe there's a lot of overlap between these different processes, and we can start to see that in the brain systems.
Thank you. And we have two more questions. Erica, you want to take this?
Sure, I can take Jacob Miller's question, which is, what about the insula do you think enables its importance for cognitive flexibility? Curious, when you brought up the functional neuroanatomy, if you had any specific features in mind, such as laminar organization, anatomical connectivity, etc., that allows the synchrony across regions and networks for flexibility.
Yeah, that's exactly the right question. We haven't. I mean, there's some groups who are looking at the laminar features, and so I won't speak to that. But I think we found some unique structural connections, for example, that I rushed past, but the dorsal anterior insula seem to have some, unique frontal connections that we didn't see for the other subdivisions. And also we've seen in the dynamics, like the dorsal anterior insula spends some time talking to all kinds of different brain regions, depending on which brain state you're looking at. The dorsal anterior insula seems to be really widespread in its interactions. So it seems, for whatever reason, to be able to interact with lots of different parts of the brain at different points in time. So there's something about its ability to interact with multiple brain regions that I think really facilitates its function across different tasks. I think, I see another question. Should I read that one?
I'll do it. So the next question is from Vikas Tarik. Thank you for such a nice and informative talk. I'm looking into the research by Kevin Stetson showing that ASD subjects could have pattern recognition, needing lots of cognitive flexibility. How does this fit in terms of your finding pointing at the rigidity?
ASD subjects good at pattern recognition. Is that the perceptual? I'm trying to think of which studies those are. Yeah, it's interesting because it's not necessarily. Oh, well, that's another question. Sorry. Okay, I was just reading what's on the screen. Yeah, and I think it's a good point. Is that there are some studies of enhanced perceptual processing in autism, for example, which is often talked about as one of the strengths in autism. So we always talk about what are the difficulties, but there's also a lot of strengths. And so, I actually am not as familiar with that literature at the moment, but it's true that. And I'm not sure if that's, I'm not really familiar with the study that you're talking about, but it's true that, we might not see necessarily deficits across the board in all different types of tasks. So, the pattern recognition, for example, is one area where we might see some strengths. But the thing that I've always wanted to make sure to note is that there's so many individual differences in autism itself. So some of the kids are showing deficits in flexibility, others are not. This changes across the life course as well. So some people become better with age or become better with different interventions. So it's true, it's not a sort of one-size-fits-all story. It's not an overall deficits across all domains. That's a good point.
I think we have time for one last question, which I can read out now, from Francisca Korb. There was one slide that got lost or was skipped involved in the IFJ dynamics. Maybe that would work now. I don't know if you're able to go back to that one. But if you're not, maybe we can share it later.
I think it's this one. For some reason, my slides were deciding which ones they wanted to show. So, was this the one? I think probably.
Yeah,
okay.
Yeah. And I can read the question that goes along with it.
Okay.
So also given that a lot of regions, like the IFJ, have been investigated and kind of related to certain functions, like implementation of a task set in the case of the IFJ, which then serves flexible behavior. How does this view integrate with your findings that the switch between brain states is a basis for this function? Basically, unfortunately, away from one brain area equals one function.
Yeah, that's a great question. This is the slide that I somehow managed to not show, but we found using some connectivity modeling approaches that the inferior frontal junction seems to be really involved in the flexible item selection per se. This is just a review summary of that study, but on the left, what the point I was showing was that even though we see these really strong activations in inferior frontal junction for flexibility of this sort, you also see those regions showing up across all manner of executive function. So if you do neurosynth meta-analysis of just the term shifting, you'll see the same kind of network regions involved as when you use the term updating for working memory, for example, or inhibition. All of these are like the same brain regions, but I think there's different ways of interactions among the nodes of the network, like the IFJ in particular for flexibility, that may be different than the way the IFJ is working in these different domains. This is just a hypothesis I'm putting out there, but I think, we often see these activation levels and say all of these brain regions are involved, which is true, but we don't know exactly the timing and the interactions between the brain regions. And this is where that concept of neural context comes in that I, introduced from Randy McIntosh, that the context in which these areas are operating in terms of the rest of the brain is also probably important for understanding the function.
All right. I think that concludes our talk for today. For anyone who has any more questions for Dr. Uddin, feel free to click on the Zoom link that I just posted in the chat for the Q&A for trainees. Thanks, everyone, for coming today.